Alright guys, let's get started. I'm your O-level edX cell biology teacher, Ayman Raman, and today we are going to be talking about chromosome, genes, and DNA, basically genetics, right? So over here, I have made a summary notes specifically for this video, where I'll be talking about the structure of a DNA, the genes, the mononucleotides, how they're interlinked, right? We're going to start all the way from like a human cell to the mononucleotide, right? And we are going to be talking about DNA replication, right? So in general, like the genetics, it's a very broad topic, right? We can discuss many things once we study about genes, right? But just for this video, we have made a short note uh, on a few contents that I want to discuss with you guys, right? So that the concept is clear. All right, so let's get started. So first of all, let's talk about humans, right? So humans, what are they made up of? Humans are made up of cells, right? And what do we get inside cells? We have the nucleus. Now if you have seen the video on cellular organization, or if you guys understood cellular organization or life processes, you would have understand by now that the nucleus, it contains chromosomes, right? So the nucleus, it contains chromosomes. So now we are going to look at this in detail. Suppose this is a chromosome, right? Suppose this is a chromosome. Now inside this chromosome, if I look within by microscope, you'll see many coiled structure, right? There will be coiled around a circular protein, right? We're going to see many coiled structure. This coiled structure is called a DNA. Now many of you would argue that a DNA is double helix. Why am I saying that it is a coiled structure and stuff like that? Yes, it is a double helix coiled structure, but you can see that double helix once you zoom in more using a microscope, right? And this coiled structure, it circles a protein, right? It circles around a protein. This protein, we call them histones, right? Hopefully I'm clear about this stuff, right? So if I look at this structure in more details using a microscope, what we'll see is the DNA. Suppose I take this part, let me highlight it. Suppose I want to look at this part. So if I extract this part using a microscope and look if I extract this part and look at this part under a microscope, what we'll see are this coiled structure, which is called the DNA, right? And the smallest segment of a DNA is called a gene, right? So again, if I look at this part under a microscope again, let's zoom into this part, right? So it looks something like this. This is what we call the gene, the smallest segment of DNA. Inside these genes are this mononucleotide that we're going to talk about, right? So mononucleotide is the smallest unit, or the structural and functional unit of a DNA, right? So this is how they are linked. Hopefully I made it clear how we end up with mononucleotide, even if we start all the way up from cells, right? If I give you guys a flowchart just for making it a lot easier, we just talked about chromosomes, right? So inside the cells we get a nucleus, and inside the nucleus we find these chromosomes, right? So if you now look inside the chromosome, what we get is the DNA, and the smallest segment of a DNA is called a gene. It is important to understand that gene is not a separate structure. It's just a segment of a DNA, all right? It's the smallest segment of a DNA. That's what we call a gene, all right? So hopefully this is clear. All right, so we already established this relationship, right? So now let's understand the structure of a gene, right? So again, let's look at a single segment of a DNA. Let's say it's somewhat like this, right? So if this was like the coiled structure and I extracted a single portion, suppose this portion, and I zoomed in a lot, I'll get a structure like this. So this contains these mononucleotides, right, that are boxed shaped with one another, right? So this substance is what we call a mononucleotide, right? These are the smallest unit or the structural and functional unit of a DNA or RNA. And this mononucleotide, this has mainly three parts. A pentose sugar, right? A pentose sugar, it's basically, pentose means 5, right? So it's a 5 carbon containing sugar. Then we have the nitrogen bases. Now the nitrogen bases are mainly, always remember, we can divide them into two types, right? Adenine, cytosine, and guanine. So these three, they are present in both DNA and the RNA. But thiamine, it's only present in DNA, right? And uracil, it's present in RNA. This is very important to remember. Thiamine, it's always present in DNA, and uracil, it's always present in RNA. And finally, we have the phosphate group. And the phosphate group, it's very important, right? Because the phosphate group, it carries a negative charge. Always remember, the phosphate group, it carries a negative charge. So this is what a structure of a mononucleotide looks like. So this is the pentose sugar, the one in the middle. Then we have the nitrogen base. It can be either adenine, thiamine, cytosine, or guanine in case of a DNA, or adenine, uracil, cytosine, and guanine in case of an RNA. Then we have the phosphate group at the other end, carrying the negative charge, right? Okay, so now let's move on. So we have already established the bases, right? So in DNA we have the adenine, thiamine, cytosine, and guanine, and in RNA we have the uracil instead of thiamine. Now what I want to talk about is the base pairing, right? This is very important. You see, these mononucleotides, these are linked with one another, right? So in DNA, the adenine always binds with the thiamine, or thiamine binds with adenine, whatever you say. In RNA, the adenine binds with uracil, and for both, the cytosine always binds with guanine. And these bonds, they're using, that's called the hydrogen bonds. We study about the different forms of bonds in chemistry, right? So we know that the hydrogen bond is one of the strong bonds, right? It's one of the stronger bonds, right? So the adenine and thiamine, or the adenine and uracil, they are linked by 2 hydrogen bonds, and the cytosine and guanine are linked by 3 hydrogen bonds, right? This is just an extra note for you guys, but always remember that they're connected by the hydrogen bonds, right? So if in general, if we talk about how these mononucleotides connect, so when talking about sideways, mononucleotide, they can connect in 2 ways, right? One is if they connect sideways, the one that you can see over here, or if they can connect up and down, right? If they connect sideways, when they connect sideways, they form these hydrogen bonds with one another. And the adjacent mononucleotides, the one present up and down, they form the phosphodiester bond. That's a link between the 2 phosphate groups, right? So this is very important to remember, that sideways they are linked by the hydrogen bonds, and up and down they are linked by a bond called the phosphodiester bond, right? Hopefully this is clear. All right. Now we have some very basic definitions that what a chromosome is. A chromosome is a thread-like structure that is only visible during a cell division. Otherwise we call them chromatin, right? The lightly visible thread-like structure. Then we have to know what gene is. It's the smallest segment of a DNA. We have to mention this term, that it is the smallest segment of DNA. Right? Let me write it a bit more clearly for you guys. Alright, and then we have to know what a gene locus is. It's just for understanding, it's not in detail in our syllabus, that a particular region in which we find a particular gene is called a gene locus. Right? Okay, what I want to discuss in detail is the difference between the structure of a DNA and the structure of a RNA. Right? By this chart, we can differentiate both of them. So DNA, its sugar is called a deoxyribose, and RNA, the sugar is called a in RNA we call it just ribose, right? Because RNA it stands for ribonucleic, and DNA it stands for deoxyribonucleic, right? We already know that nitrogen bases, that in DNA the nitrogen base is thymine, but in RNA the nitrogen base is uracil. The remaining are the same, right? In DNA, base pairing occurs where adenosine binds with thymine, right? Adenine binds with thymine, and in RNA, the adenine always binds with uracil. Finally, the DNA is double-stranded. Let me draw this clearly. So the DNA, it's double-stranded, right? But the RNA, it is a single strand. Okay, so it's important to know the location of this DNA and RNA, right? So we already know that DNA, this is present inside the nucleus, or inside the chromosomes, right? But the RNA, a good example of its location is in the mitochondria. It's even present in the cytoplasms, right, or the ribosomes. Right, so this is very important to remember. Okay, DNA, it has no subtypes, but RNA, it has 3 subtypes. Right, and these 3 subtypes, these are very, very important in protein synthesis. We are going to talk about them in way more details during our lecture on protein synthesis. Right, over here we just need to know the names. That is the messenger RNA or the mRNA, that basically forms an exact copy of the DNA code, right? Then we have the transfer RNA, right, which helps to carry the amino acids from the cytoplasm to the ribosomes. And finally, we have the rRNA or the ribosomal RNA that forms the part of the ribosomes. Right, so this is very, very important in our syllabus to know that how many types of RNA are present and what's their function. Right? Okay, now the main thing that I wanted to talk about in this video or in this lecture is the process of DNA replication. So DNA replicates, right? When a cell divides, the DNA replicates, so one can form into 2, right? So we have to understand this process, right? And before understanding the process of DNA replication, we have to understand the name of 3 enzymes in very much details, right? We are going to talk about 3 enzymes. Always remember the name of these 3 enzymes in case of DNA replication. DNA helicase, then we have the DNA polymerase, and finally we have the DNA ligase. Alright, now let's understand the process of DNA replication. When I'll explain this process, I will also state the function of the enzymes. Alright, so suppose this is the DNA, right, that we want to replicate. Now this DNA, it contains, if we zoom into a portion, right, it contains the double strand, let's say an adenine with thymine, a cytosine with guanine, a guanine with cytosine, right? This double strand contains this. So we have to break the bond between these strands, right? Suppose we have to break these bonds, right? And these bonds are broken by the enzyme DNA helicase. So we already discovered the function of one enzyme, DNA helicase. This breaks the hydrogen bond, right? Okay, so now that the hydrogen bonds are broken, we can say that the two strands are now separated, right? The two strands, they are no longer connected to each other, right? So they're separated. Now inside the nucleus, this is all happening inside the nucleus, right? Because we already established that the nucleus, it contains the chromosomes, and inside the chromosomes it contains the DNA. All right, so we already established that, right? So inside the nucleus there are many free mononucleotide present, right? So inside the nucleus there are many free mononucleotide, and these mononucleotide, these will pair up along these free strands to make new DNA, new strands, right? So if I draw again, so suppose this was adenine, cytosine, and guanine, a new strand will develop along this single strand. Suppose the thymine, guanine, and cytosine, right? And they will form a hydrogen bond back, but this time they will use DNA polymerase, right? But remember, at the beginning of the lecture, what did I say? That you have to form a strand sideways, you have to form a bond sideways, that is done by the DNA polymerase, but you also have to connect them up and down, right? The adjacent mononucleotide. So over here the adenine, it needs to link with cytosine, right? So over here the thymine, it needs to link with guanine, right? So this is done by a bond called the phosphodiester bond, right? We already discussed this part. But this phosphodiester bond, this is being made by the DNA ligase, right? And this is also happening to the other strand, right? So we talked about only this strand, but this is also occurring, the same thing is also occurring for this strand as well, right? So over here we can state that the DNA polymerase, it forms new hydrogen bonds between the adjacent mononucleotide, and that the DNA ligase forms new phosphodiester bond between the adjacent mononucleotides, right? And this is how ultimately two new strands are formed. So from this one strand of DNA we are getting, so first we made single strands, right? How did we make single strand? With the enzyme DNA helicase. Then the single strand, these are being converted back to the double strand. Let me use a different color just to show the formation of the new strand, right? So this is the new strand that was formed by two enzymes, right? What are the enzymes? The DNA polymerase and the DNA ligase. So this is the process of DNA replication, right? And this is very, very important to understand, right? We have three enzymes in details, right? You can see that the DNA helicase, the DNA polymerase and the DNA ligase. Okay, so this is the process of DNA replication, and this is very, very important in genetics, right? In the whole topic of genetics for our biology O level, we have two very, very important contents. One is the process of replication, and another is the process of protein synthesis, right? There is only one difference over here. In replication, the DNA strands, these are broken completely, right? The hydrogen bond that is present between each mononucleotide, this is broken completely by the enzyme DNA helicase. But in case of protein synthesis, we only break a small portion, right? We will discuss that once we talk about the protein synthesis in detail. But my goal over here is to make you guys understand the process of DNA replication. Alright, so just to give you guys a summary of the process of DNA replication, what did we do? Mainly, we have a DNA strand, right? This DNA strand, it is broken down completely by an enzyme called the DNA helicase, right? Always remember, in our O level, they do ask the function of these three enzymes, whether it's in the multiple choice or they give a 1 mark question, but they do ask the function of the three enzymes, right? So we talked about DNA helicase that breaks the hydrogen bonds between them, right? So once the hydrogen bond is broken, we can see that the two strands are no longer together, that the two strands are now separated, right? Okay, then once the two strands are separated, there are many free mononucleotides present inside the nucleus, and what these mononucleotides do is that they align themselves across their complementary base pair. This term is important, that they align themselves across their complementary base pair. Complementary base pair. What does this mean? It means that, see, the mononucleotides are fixed, right? So if we have like a molecule of or a mononucleotide cytosine, and adenine would never pair with it, right? So an adenine would never line up along a cytosine molecule. It would always wait for a guanine molecule, right? Oh, sorry. This will always wait for a guanine molecule, right? So similarly, when there is a thymine, it will always pair along a adenine molecule, right? So this is what we mean by the complementary base pair, that they wait for their complementary mononucleotides, and once these complementary mononucleotides join, they join by sideways and up and down, right? How do they join sideways? They join sideways by a bond called the DNA hydrogen bond, and this is formed by the DNA polymerase, right? So this DNA polymerase, it was forming the hydrogen bond. Similarly, up and down the mononucleotides, these are connected with each other by another bond, right? What is that bond? That's the phosphodiester bond, right? So they're connected up and down by this phosphodiester bond, right? So this is the process of DNA replication. Alright, so we already talked about the whole process of DNA replication. We talked about the each and every step, and we also talked about the enzymes. Again, this is very important to note the function of three enzymes. They are mainly going to ask the functions. Okay, finally, let's move on. Okay, now for our final topic over here, we are just going to talk about the genetic codes, or the triplet codes. What is a code? Always remember, a code, alright, or what we call a codon, is a 3-pair codes or format. Let me write it more clearly. It's like a 3-pair format to represent a sequence of base pair, right? So a good example would be, for example, let's say ATC, right? or let's say GTC. If we have like a DNA strand that contains A, T, C, G, T, C, then suppose C, C, G, we cannot write it like this. We have to put spaces between them, right? That's how we represent the codes, right? So if I want to represent it, we have to write it like this: A, T, C, and we have to give a space and write G, T, C, and then again C, C, G, right? So a codon is basically a 3-pair code to represent the base pairs in a DNA strand. Alright? So we call them a codon or a triplet code. Alright? Whatever you guys want to call them. And always remember, this is very, very important, that over here we have 20 basic amino acids. But these 20 basic amino acids, these can be represented by 64 possible codons. So by this thing, you can understand that that one amino acid is represented by more than one code, right? So suppose that the ATC or GTC, they both can code for the same amino acid, right? It can be, for example, valine. This is an amino acid, right? So they both can code for these amino acids. It's not necessary that each amino acid would have a single code, right? But one codon, it cannot represent two amino acids. For example, ATC will only represent valine. It cannot represent another amino acid, right? One amino acid can have more than one code, but one code cannot represent more two amino acids at the same time. Alright? This is very, very important to understand. Okay? So in this video, or in this lecture, we mainly talked about the structure of the gene, mononucleotides, DNA. So we talked about the basic intro to our genetics, right? We talked about in detail the structure of mononucleotides, right? The parts it has, the ribose sugar, deoxyribose sugar, right? That is the main form of pentose sugar. Then we talked about nitrogen bases and the phosphate group, again giving importance to the negative charge on the phosphate group. We talked about their bonds, that they form the phosphodiester bond and the hydrogen bond. And finally we talked about the differences between the DNA RNA, the process of DNA replication in detail, right? Talking about the 3 enzymes, right, and their functions, and the genetic code, right? So this is the introduction to genetics.